So, in the last class, we have tried to understand about the neo-Darwinism, or which is also popularly known as synthetic theory of evolution. It's a not a new theory, but I can say that it is the modification to what the Darwin and Wallace has suggested. Because when Darwin and Wallace has proposed the theory of natural selection, they were blissfully ignorant of many things, especially in the modern genetics, molecular biology, and even paleontology was also not fully grown branch at that time. So, based on all the inputs, uh, the followers of Darwin, T.H. Huxley, and others, uh, have proposed a lot of things and included many things in this theory of natural selection, and that is popularly known as synthetic theory of evolution, or sometimes it is also called as neo-Darwinism. So, in the last class, we have tried to understand about the different factors which have contributed to the uh, evolution of this theory of synthetic theory of evolution. So, in that, natural selection is a part, and they do not deny the role of natural selection in the evolution, but along with that, the main concern of the synthetic theory of evolution is how the variation is brought, because even Darwin has failed to talk about the origin of variation. Though he has identified the importance of variation, he has failed to understand or explain the origin of variation. So that's why so many other inputs, starting from mutation, or it may be gene flow, gene pool, genetic drift, recombinations, and even isolations, all those things have been included in this. Now, uh, in the synthetic theory, after knowing the background, we have tried to discuss about the different uh, aspects of that. In that, we are discussing mainly again the natural selection, because that is the key factor, uh, or it is the central dogma of even the theory of a synthetic theory of evolution also. So, in the last class, we have tried to understand how the natural selection operates, and today, for a few minutes, we will continue with the natural selection only. Then we will certainly move on to the study of different other factors which contribute to the origin of variation, and in the end again we will discuss this theory of natural selection once again. Now, in the last class, I have told you about the different uh, characteristic feature, or the main points or highlights of the natural selection. Now, first I will discuss about the type of natural selection, then move on to uh, the factors which contribute to the variation. Now, when we talk about the natural selection, there are three or four different types of selection which we observe in nature. One is the stabilizing or balancing selection. What do you mean by this stabilizing or balancing selection? This type of selection is also observed in the nature, where extremities are avoided. Extreme varieties from both the ends of the frequency distribution are eliminated. In that, what does it mean? It means that natural selection doesn't generally prefer the extreme variations, but then what it prefers is average or normal variation, which is slightly different from existing. Suppose if all of a sudden, if there is a extreme variation, generally in this stabilizing or balancing selection, that has been avoided. So, as a result, natural selection in stabilizing selection or balancing selection is a bell-shaped curve. See, these are the extremes in one hand, and these are also extremes in the other one. This is the original population. So this, if you see this balancing selection, it prefers only those variation within that limit, and average variations are preferred. This extreme variation this side or this side, they are eliminated. For example, if you take the example of sickle cell anemia is seen and it is a stabilizing or balancing selection only in those population where the malaria is very very prevalent. Only in this area the sickle cell anemia has certain uh, role where it enhances the survival value of the individuals, but not in other cases. So that's why selection in the both the this one in this area, whatever the variation is there, it is selected, but not the, so the extreme variations, extremities are generally avoided. This is what is known as stabilizing or balancing selection. Yeah, in the very beginning, I would like to clarify one more point. When we talk about natural selection, this stabilizing or balancing selection is not the only type of selection. There are different type of selections also. But this kind of selection is one type of natural selection which prefer the average one or the normal one, not the extreme values. Then if you go to the next one, this is what you call the directional selection. This is not like the stabilizing selection. So the population changes towards one particular direction along with the change in the environment. When there is a change in the environment, the population, if it wants to survive, means along with the nature, it should also change. I have already told you the nature will select only those organisms which are compatible with it or which are adapted to the changed environmental conditions, because the nature is always dynamic, not static. So when the nature evolves, if the population which is living in that nature, if it has to survive, means along with the change in the nature, the organism also should show the changes which makes them more compatible with the changed scenario. So that's why the population changes towards one particular direction whenever there is a change in the environmental conditions. So here what happens is individuals at one extreme is eliminated while the individual at the other extreme is favored. See, this is the normal population. This is the normal original population which you call here. But what has happened is the nature has changed. So correspondingly, this new population after selection will also move in one direction, leaving all these populations behind, because it has preferred those characters or those individuals which have some extreme changes, but in one direction, which will favor them to become more fit to the changed condition of the environment. For example, the industrial melanism and DDT resistance in mosquitoes is a good example for directional selection. In case of the industrial melanism, you have seen in case the pepper moth. Earlier, the entire population, most of the population was gray colored, but after the industrial revolution, it changed from gray color to maximum number of the individuals changed into dark color, because it was very necessary to survive, and it has to adapt with the changed nature. Even the DDT resistance in mosquitoes is also a good example. The ones, though now the DDT is banned because of its poisonous effect, because it's not a biodegradable pesticide, now it is banned. But in olden days, this DDT was uh, extensively sprayed everywhere in order to control the mosquito. It was considered as a wonder pesticide, and it is the cheapest pesticide also. It was the what has happened when the DDT was sprayed, most of the mosquitoes died, but only few could survive because of some their better genetic makeup, where DDT has no uh, effect on them. Especially the DDT used to affect on the nervous system of these mosquitoes. But most of the mosquitoes died because of the spray of the DDT, whereas few survived because of the some better genetic makeup where the DDT showed no effect on them. So as a result, what has happened after the spray of this DDT continuously for few years, all those uh, mosquitoes died, but only those mosquitoes survived which are having the better genetic makeup, and they multiplied and passed this character to their offspring. As a result, a day has come, even if you spray did it uh, they were not knowing about this what you call the genetics and molecular biology, crossing over, recombinations, all those things they were not knowing. So that's why uh, they fail to explain the origin of variation, but they have identified the importance of variation. Now, in the next one or two classes, we will be discussing about how the variation is brought and what are the factors which bring the variation. In this, the first thing we will start with recombination. What is recombination? Recombination, which all of you know in your PUC also you might have studied that, is a process by which two DNA molecules exchange the genetic information, or where the genetic information is swapped between the two DNA molecules. As a result of this, there is a production of new combination of alleles. This is what we call the recombination, where it happens during the crossing over. Especially uh, this recombination creates the genetic diversity at the level of genes and thus cause the variation. And all of you know, may be knowing, this is the reason, though the offsprings resemble their parents, they are not exact copies of the parents. You may be having some traits from your father, some traits from your mother, and apart from that, you may be having your own individual uh, specific traits which are not seen either in your either of your parents. So this is because of the recombination, and that brings about what you call the variation. So here, the variation in eukaryotic cells, especially the recombination occurs during the Pagetian stage of meiosis 1 in the substage of meiosis 1, which all of you know. There's a recombination. When the crossing over takes place, then it will happen. But in case of the eukaryotes, sometimes may also occur during the mitosis, where two um, chromosomes, two sister chromatids separate after the chromosomal replication. So this swapping or exchange of the segments of the DNA during the crossing over, what you call recombination, brings the variation. There are different types of recombination. One is what we call homologous recombination. So here in the homologous recombination, the nucleotide sequences. What are nucleotide sequences? Adenine, guanine, thymine, and cytosine. These are the nucleotide sequences. They are exchanged between the two similar or identical DNA molecules, where they are exchanged. Undoubtedly, the nucleotide sequences are exchanged, but it is between two similar or identical DNA molecules. See here, this is a these are two homologous DNA segments, and the exchange is in between them only. See, the crossing over is in between them. These are identical almost. Mostly used by the cell. When it happens is when the cell tries to repair or the harmful breaks that occur in both the strands of DNA, in the especially DNA double-stranded break. When it happens, if there are any breaks, and if the during the proofreading mechanism, if they identify there are some repair is to be done, then it is done. So here what happens, as the change is the recombination is between two similar or identical DNA molecules, though the recombination takes place, but no net gain or loss of nucleotides. Same nucleotides will continue. So the though it brings the variation, but the amount or the intensity of variation is very very less, as the exchange of these nucleotide sequences have taken place between the two similar or identical DNA strands. Then second one is non-homologous or illegitimate recombination. So this happens between two non-homologous uh, DNA. So here, where no large-scale sequence similarity is apparent, there is no similarity. See, this is one ABC, where the nucleotide sequence are like this. This is another DEF. These are the nucleotide sequence on two different DNA strands, and the recombination makes it a part of this strand is attached here, whereas uh, as part of this is attached to the next one. So here the sharing is between two non-homologous DNA molecules which are not similar or identical. So as a result, whatever the variation that has been brought is certainly more uh, intense, or in other words more visible also. Then the third one is site specific recombination. Uh, this occurs particularly short sequences of the DNA strand, which is about 12 to 24 kilobytes. It takes place in the segment of DNA strands, and usually processing only limited degree of sequence homology. See, this is where you can see this is the E. coli uh, DNA strand. See, there's an enzyme by name integrase. It helps and bind this lambda here in between these two. So this is what you call site-specific recombination. Only in this region, this has included a new sequence or new set of nucleotide that has been named as lambda. Then the fourth one is replicative recombination. This recombination which generates new copy of a new segment of DNA, which happens during the DNA replication. Uh, just you recollect what you have studied in your PUC with respect to this DNA replication. It's a type of recombination, and it generates a new copy of the segment of DNA, because the parents one strand function as the template, and on that a new copy of the DNA has been formed. See, this is the old copy ABC, and the other copy which is formed is also exactly same. And here what happens, they are replicative recombination. Here also the amount of variation brought is very minimal. Then the reciprocal recombination. What do I mean by this reciprocal recombination? Where equal exchange of genetic information. Whatever the amount of genetic information that is shared or swapped between the two DNA strands is exactly equal. And as a result of this, what happens? The new DNA strand or molecule carries the genetic information which has been derived from both the parents. So, exactly the same amount of DNA has been swapped between the paternal and maternal chromosome. That is resulting in a DNA molecule that carry the genetic information which is derived from both the parents and in equal amounts. So, as a result, the number of alleles remain same. Or what happens is only their arrangement has been changed. Their arrangement may be uh, changed, uh, but the number of alleles that remains same. Then the last one, the gene conversion, which is also known as non-reciprocal recombination. Here it is the one-way transfer of the genetic information. One way, either from paternal nucleate DNA to maternal, or converse is also true from maternal to paternal. One way transfer of genetic information. As a result, what happens? The number of alleles will change. Uh, Transfer of genetic material from a donor to a highly homologous acceptor sequence. Okay, here there is one limitation. It cannot transfer to any of the DNA strands or DNA strands, but it should first identify highly homologous DNA strand, and then it can transfer the genetic information from donor to recipient or acceptor, but in a highly acceptor sequence it should be. Uh, Now, what are the advantages of this uh, recombination? We have discussed different types of recombination. What is the advantage? One is, all of you know that the main advantage of the recombination is to enhance the genetic diversity in a population. Because of any type of this recombination, new gene or allele combination takes place. Because there is a exchange, swapping or unidirectional flow of the genetic information from either from paternal to maternal or maternal to paternal or vice versa. So this enhances the genetic diversity in a population. Here, another advantage is negative selection can remove the deleterious alleles from a population without removing the entire chromosome. This is the most important thing in any population, especially if the population wants to stabilize. In any given population you take, there are uh, characters or traits which are favorable, which are not favorable. Uh, Sometimes they may be harmful also. But here, when there's a recombination, there's a chance for the nature to eliminate those deleterious or those characters by negative selection. That means not by selecting them. So as a result, it is not removing the entire chromosome, only a small fraction of the DNA where a particular gene which is not favorably situated that can be eliminated and thus makes the population more fit, entire population. Then it ensures the proper segregation of homologous chromosome. uh, all of you know the homologous segregation of the homologous chromosome is the first step in any of the recombination. So this ensures the segregation, proper segregation of the homologous chromosome. Homologous means the chromosome which are similar but not identical. Thus, it ensures the genetic stability of the organism. The main purpose of this recombination is to establish the genetic stability uh, by eliminating the less favorable genes and uh, promoting the more favorable genes, thus attaining a genetic stability of the organism. Especially here, the site-specific recombination systems. They are the most potent modifiers. They can modify most of the time instead of uh, where there is no effect, but whereas here the site-specific recombinations, they can uh, eliminate certain traits and induce new traits. So that's why they are the most potent modifiers. And it is used to map the genes on the chromosome as recombination proportional to the distance between the genes. Of course, this is if you know the recombination, you can map the genes on a chromosome also. Uh, and it is also used making transgenic cells and organism. Transgenic organism where the uh, genetically modified one, transgenic cells or organisms can be formed because of this recombination technique. Nowadays, extensively this recombination technique has been used using the biotechnological tools, so it has a lot of advantages. But at this level, where we are interested is it brings the new allele or new combination, and it brings more and more variations. That's more important for us. So this is how the recombination brings the uh, variation. So that's why the recombination is considered as one of the very important tool for the origin of variation. Like recombination, another tool for variation or another uh, method how the variation is brought about is mutation. What is mutation? All of you know that sudden heritable changes in the genetic material or character of the organism, we call it as mutation, sudden and heritable, because this is very, very important. Remember, we used to, in theory of natural selection, when it was proposed by Darwin and Wallace, they used to say that evolution is a slow process, but it's a continuous process, and it is a process selected by the nature. So now we are not ruling out the natural selection, but we are talking about the variation, and variation is the tool for natural selection. But this variation can need not be very slow process. Sometimes it can be equally fast. This is what uh, uh, the neo-Darwinism or synthetic theory of evolution justifies, and how it can justify using this mutation. Mutation, as a name itself, definition, the very definition says that it's a sudden heritable change in the genetic material or the character of the organism. So the individuals which are showing this mutation, they are called mutants. And there are many agents which causes the mutation, they are called as mutagens. Mutants is the individual which shows the mutation, and the agents which are responsible for bringing the mutation, they are called as mutagens. In mutation also there are different types. Basically we can have two types. One is spontaneous mutation. Spontaneous, or it's a natural mutation, because it occurs naturally without a known cause. Even the scientists have failed to identify the cause for the mutation. It may be because of some errors in proofreading during the DNA replication, or various factors we can attribute to it. But it occurs naturally without a known cause. That is what we call spontaneous mutation or natural mutation. The second one is induced mutation. This induced mutation is the result from exposure to mutagens. When an individual, when I say individual, it is their chromosome or the genes which is exposed to some mutagenic agents. What are the mutagenic agents? One is ionizing radiation, or ultraviolet rays, or various chemicals which can bring the mutation. So this is what you call induced mutation or artificial mutation. Uh, that can also cause the sudden heritable change in the genetic material or in the character of the entire organism. So that type of mutation we call it as induced or artificial mutation. This is based on the type of mutation. Then even we can have different types of mutation based on the tissue of origin, tissue or cells of origin. One is the somatic mutation and another one the germinal mutation. What do you mean by somatic mutations? That occurs in the somatic cells. Somatic cells are the body cells which do not have any role in the sexual reproduction. So but in asexually reproducing species, somatic mutations transmit from one parent to next progeny. So that's why these mutations can have the effect. But in sexually reproducing organism, this somatic mutation has no role to play. But in asexually reproducing species, if it even the mutation occurs in the somatic cells or the body cells, though they are not taking part in the sexual reproduction, but here it is the asexual reproduction that this mutated character may get transmitted to the offsprings or to the progeny. Then the next one is the germinal mutation, which occurs in the gametic cells, in the gametes, male and female gametes. And all of you know that these gametes take part in the sexual reproduction, and if the mutation happens in this gametic cells, that's the uh, testis, sorry, that's in the sperm and egg, then uh, if they contribute to the sexual reproduction, then naturally these mutations or these new traits or new characters are being transmitted from one generation to the next generation. So in a sexually reproducing organism, if the mutation should show effect, means it should happen in the germinal mutation or in the gametic cells. Then another classification of the mutation is based on the direction of the mutation. One is forward mutation. This occurs from normal type to mutant type in one direction. It will carry the population in forward in one direction. That is what you call forward mutation. If it occurs in the normal type, and as a result a mutant allele is formed. That is what you call the forward mutation. The second one is the reverse mutation, reversal of the first one, where the mutation occurs in a mutant allele. Mutation occurs in a mutant allele and brings it back to the normal type. That is the reverse mutation. So these are the two types of mutation based on the direction. One is forward mutation and another one is reverse mutation. Now, the most important classification of the mutation is the chromosomal mutations and gene mutations. Let's concentrate more on this chromosomal mutations. What is chromosomal mutation? If the change occurs in the number or structure of the chromosome, if the change takes place not at the subtle level but at the gross level, occurs in the number and structure of the chromosome, that is called as chromosomal mutation. It should happen in the chromosome. A part of the chromosome or the number of chromosome may change. That is known as the chromosomal mutations. So the first one is in this chromosomal mutation is the chromosomal aberrations. What do you mean by aberration? This refers to the morphological changes, change in the appearance of the chromosome, morphology of the chromosome, without affecting their number. The number will remain. If at all, if the chromosomal aberration happens in a human being means, in human beings we have 46 chromosomes, that is 23 pairs of chromosome. That number is not going to change. But a one chromosome somewhere in the 21st pair chromosome or somewhere, there may be change in the morphological changes in the chromosome, where the chromosome number is not going to affect. Number remains same. In these chromosomal aberrations, there are four different types of aberrations can happen. One is deletion. What do you mean by deletion? A block of a gene from the chromosome may be deleted. This block may be at the terminal end of the chromosome or in the intercalary also. Wherever, if there's a small part of the chromosome having a gene block, if it gets deleted from that one, as a result, what happens is that arm or that chromosome becomes shorter. There's a change in the morphological structure. That is what you call deletion. Second one is duplication. An extra block of genes may be added to the another chromosome. Already that block may be present in that chromosome, but same copy of the same block has been added to this from a deleted chromosome generally. The presence of some genes more than once as a result. Then you can have addition, deletion, or addition or duplication, or even you can have translocation. In the translocation, I have not, somehow that slide is missing here. Translocation, a chromosome may turn 180-degree and exchange the segments within that, or it may lose. That is what you call translocation. So such morphological changes which occur in the chromosome, we call it as chromosomal aberration. Like chromosomal mutation, we also have gene mutation. This is at the subtle level. I have told you The chromosomal mutation is at the gross level, whereas the gene mutation is at the subtle level, at the level of genes. Many times it goes unnoticed, invisible changes when it happens in the chemical nature of the genes. And in most of the time, the gene mutation, see, chromosomal mutations, they undoubtedly show certain variations. Whether it is positive or negative, it's a totally different one. But these gene mutations uh, need not produce any detectable morphological effect for the very simple reason that most of the gene mutations are recessive mutations. Suppose if the dominant gene mutation happens in a homozygous condition, it may produce a phenotypic change. But generally, most of the gene mutations are recessive, so that's why they fail to show any pronounced phenotypic changes. Only gene chemistry may change. The gene mutations, there are three types: frame shift mutation, substitutional mutations, and point mutation. What do I mean by this frame shift mutation? So, here the mutation happens undoubtedly in which the reading of the frame of base sequence shifts laterally, either in a forward direction due to insertion or backward direction due to deletion. See here the framework deletion, where you can see this is the mutation in presence of messenger RNA protein. See what has happened. This has changed. This is the frame shift mutation where you can see the forward changes. Sometimes it can be backward also, where one or more nucleotides are lost from the segment. In insertion, one or more nucleotides are added. The same to this, see, this has been added. One or two nucleotides are added in the segment of a DNA representing the gene, where in deletion, one or more nucleotides are lost from the segment. This is deletion. There are two types, the frame shift mutation. Then the second one is the substitutional mutation. In this substitutional mutation, the nitrogen base of the nucleotide sequence is changed with another one. How? To give an example, in sickle cell anemia, the glutamic acid of B chain of hemoglobin is replaced by another amino acid, that is the valine amino acid, to form a detectable defective hemoglobin S. From hemoglobin A, it is changed to hemoglobin S because there is a change in the uh, glutamic acid, which is a which is a amino acid. Glutamic acid is replaced by the valine amino acid. So here substitutional mutation, there are two types. One is the transition. If the purine is replaced by the purine and pyrimidine is replaced by the pyrimidine, you know that in a nitrogen base there are two types, purines and pyrimidines. If the purine is replaced by a purine, if the pyrimidine is replaced by a pyrimidine, it is called as transition. The other one is transversion. In this transversion, purine is replaced by a pyrimidine or pyrimidine is replaced by a purine. Then if such changes happens, that is known as transversion. And the third type of mutation is point mutation. Here, in the point mutation, a change in a single nucleotide, only one nucleotide changes. Here there are two types. One is synonymous mutation, where a codon with another codon that codes for one amino acid, so that produced amino acid sequence are not modified. One codon is replaced by the another codon, but that also code for the same amino acid. That is what is known as synonymous mutations. As a result, amino acid sequence is not modified because the same amino acid has been produced by another codon. That is the software for, that the protein synthesis. Then non-synonymous mutations. This is more important, where one codon is replaced with the other codon, and that codes for different amino acids. One codon is replaced by another codon, and that codes for a different amino acid. Instead of one amino acid, another type of amino acid is formed. As a result, the amino acid sequence is altered. That is non-synonymous mutation. Here, there are two types. One is missing mutation, where a change in the nucleotide causes the substitution of different amino acid. It causes the addition or substitution of different amino acid. Uh, this can render in the resulting of the protein non-functional. As a result, all of you know that the basic unit of the protein is the amino acid. And if the amino acids, different amino acids, if they are arranged in a specific sequence, it will result in a functional protein. But when there is a change in the amino acid pattern, it may result in a protein which is non-functional. That is missing mutation. Another one is nonsense mutation. This is in a very rare cases it happens. Point mutation results in a premature stop codon or a nonsense codon. So in codon there are different types. Some are stop codons, start codons, nonsense codons. In when there's a transcription of messenger RNA, it is truncated of a non-functional protein product because uh, prematurely the stop codon comes and protein synthesis stops, or nonsense codons interfere with that. As a result, the different types of uh, non-functional protein products may be there. This is the DNA level. This is a silent one. That is the protein level here. Here nonsense, what happens is a stop codon will there and no synthesis of protein. And here a missense mutation. See here, instead argin has been formed and it will change uh, the useless proteins are produced here. So this uh, recombination or mutation brings the genetic variation. This is the reason how the genetic variation is brought about. And in fact, all of you know this genetic variation is a tool for evolution. So ultimately, these genetic variations among the individual, it is caused by the different in genes or DNA segments, which all of you know, because these that too in the mainly in sexual reproducing organism, if the genetic variation happens in the gametes, then only they can bring the differences in genes or other DNA segments. And phenotype, that the physical expression, is the product of inherited genotype. It is the inherited genotype. Phenotype is the product of inherited genotype, and to a certain extent, the environmental influence is also there. In a certain level, there is an environmental influence is also there. So, this the product inherited genotype and environmental influences uh, are responsible for the phenotype. Then, when a natural selection can act upon the variation with a genetic component. Without genetic component means even if the natural selection selects it, it is not going to perpetuate. It is not going to participate in the offspring formation. So that's why natural selection can act only on the variation with a genetic component. Otherwise, it's of no use. See here, these are the certain this thing. This see, this is a caterpillar. This is a caterpillar. Which one has the better? This is one variation. This is another variation. See, this caterpillar can be easily identified by the predator because it is in a this color. All these caterpillars, they are eating the green leaves. In a green environment, they can be easily spotted. But whereas here, see, they are the gray colored. The stem is also gray colored and they are hardly identified by the predators. So you one expect the variation within a population. It's a must. Any population you take, there is a variation. Both discrete and quantitative characters, quantitative, they contribute to the variation within a population. And these discrete characters, they are classified on either or basis, or quantitative characters vary along the continuum with the population. So this I need more explanation either on basis or continuum population. Uh, I feel probably I can uh, take it in the next class only because there is time is not permitting me because at 12:30 I have one more class.